children, last time we just have read about the properties of the DNA, and I told you about the two main properties of the DNA. That means that DNA undergoes the process of autocatalysis and it also undergoes the process of heterocatalysis. During autocatalysis, it makes its own copy. That means from DNA, a DNA molecule is made, and during the process of heterocatalysis, from DNA, a RNA molecule is being made. So today what we will be studying is the protein synthesis, and under this protein synthesis we can see the property of the DNA, that is it behaves as an heterocatalytic molecule. But before going into that, let us talk about the protein synthesis. So information for the protein synthesis, that is present in the DNA only, but from DNA it needs to be carried away by a specific molecule, then to the cytoplasm, and where ribosomes are there and the proteins are being synthesized. So this overall concept, this was given by a scientist named Francis Crick. He proposed a dogma, and that dogma was termed as central dogma. Now when we talk about the central dogma, if we give it in a pictorial representation, central dogma is basically from DNA, RNA is being synthesized, and from RNA a polypeptide is being synthesized. So in short, when from DNA and RNA is being made, the process is termed as transcription. The process is termed as transcription. Once again, when from DNA and RNA is being synthesized, we say that this process is known as transcription. And when from RNA a polypeptide is being made, this process is termed as translation. So this overall, this process is termed as central dogma. So in central dogma, from DNA to RNA and from RNA to polypeptide. So let's read these, or let's check these steps in detail, that what happens during the process of transcription, what are the enzymes required, or what are the prerequisites for the transcription, and what are the prerequisites for the translation. Now let's start. Now suppose this is a DNA segment, and this DNA segment, as you know, that this DNA segment contains heterochromatin also and euchromatin also. Heterochromatin is basically that part of the DNA which is not transcribed, and euchromatin is that part of the DNA that's being transcribed. Suppose we take this part of DNA and we say that this is the segment of DNA. We say this is the DNA segment. This is DNA segment, and this DNA segment is being transcribed into mRNA, and that mRNA makes a polypeptide. So this segment of DNA that is capable of being transcribed into mRNA is termed as gene. So overall, if we define the gene, we say gene is the functional, we say that gene is the functional unit of inheritance. We say that gene is the functional unit of inheritance. Now, apart from that, we say the segment of DNA, the segment of DNA that makes tRNA, that's the transfer RNA, and rRNA, that's ribosomal RNA, is also termed as gene, is also termed as gene. So when we talk about the gene, we say that gene is the functional unit of inheritance, and the segment of DNA that makes tRNA and RNA is also termed as gene. Now there is also one term which we call as cistron, Now cistron is the segment of DNA that synthesizes a polypeptide. So today what we will do is basically we will read about this cistron and how this cistron is capable of synthesizing a polypeptide. Let's start. So starting the process of transcription. So what is transcription? Transcription is basically the information that is present in the DNA. It is copied from the DNA to mRNA. So transcription is the process of transferring the information present on the DNA to mRNA, or simply the synthesis of mRNA from DNA template, or the synthesis of mRNA from DNA. And this property from where the mRNA is being synthesized from DNA, this property is also known as heterocatalysis. So this property is also termed as heterocatalysis. So when we elaborate this transcription, we can see as I told you, in case of eukaryotes, the DNA is a long linear molecule and it is not active throughout. In this DNA, you can see there are certain units which can transcribe and there are certain regions which cannot transcribe. As I told you, the regions which can be transcribed into mRNA, they are termed as the euchromatic regions, and which cannot be transcribed, which are transcriptionally inactive, that region is termed as heterochromatin. So if we make a DNA strand, suppose this is a DNA strand, and this DNA strand, this is the polarity, this is the second strand with the polarity 5', 3'. Now if this region is to be transcribed, that means if this region contains the information in the form of the genes, so this region, this whole region needs to be transcribed. So when this region needs to be transcribed, it forms a unit that is known as transcriptional unit. So when we, if this is the, once again, if this is the DNA segment that needs to be transcribed, or we say this is the segment of DNA that contains the information for the polypeptide synthesis. So, this region, we can say this region is termed as transcriptional unit, because this region or the information that is present in the strand needs to be transcribed in the form of the mRNA. So, when we say this region basically so when we see this region that needs to be transcribed during the process of the transcription, we say this region forms a transcriptional unit. So, we say this region forms the transcriptional unit. So, remember, this transcriptional unit is present on that DNA molecule, and this has the information for the protein synthesis. As you know, for the protein synthesis, the first step is the transcription. Now, during the process of transcription, the unit that needs to be transcribed is the transcriptional unit. When we talk about this transcriptional unit is divided into 3 areas. So, this transcriptional unit has 3 areas. When we talk about the areas of the transcription, we say this region, this is known as the promoter. This is known as the promoter, and this region is termed as the structural gene. This is termed as the structural gene, and this region is known as terminator. This is known as terminator. So, talking about the transcription unit, now as I told you that this transcriptional unit is composed of 3 parts. So, first part is known as the promoter, and the second part is termed as the structural gene, and the third part is known as the terminator. So, as you know, there are the 2 strands here in this transcriptional unit. First strand is this strand. As you know, this strand, we call this strand as coding strand. This strand is termed as coding strand or sense strand, or this is also termed as complementary strand or complementary strand or complementary strand. And this strand, which has a polarity 3-5, this is termed as the non-coding strand. This is the non-coding strand. It may be also termed as antisense strand. Antisense strand. Or this strand might be also known as, or this is also known as template strand or template strand. This is termed as template strand. So, there are 2 strands involved there in the transcriptional unit. First strand is having the polarity 3-5' and second is having 5', 3'. And in this strand, this strand that is having the polarity 3-5' is used as an template strand. Just remember that in DNA replication, both the strands were utilized. That means this strand were also utilized and this strand was also utilized. Now, when we talk about the transcription, during the process of the transcription, only one strand is being transcribed, and that strand is basically the non-coding strand is being transcribed. Let me tell you why this we call it as an non-coding strand. Now, as I told you that this strand is being transcribed which has the polarity 3-5'. So, when we talk about transcription, transcription both takes place in prokaryotes and in eukaryotes. When we talk about transcription, so transcription takes place both in prokaryotes and eukaryotes. So when we talk about prokaryotes, there the process of transcription requires certain enzymes. So the enzymes which are required for transcription of the prokaryotes, that is known as RNA polymerase. That is RNA polymerase, or we also call it as DNA dependent RNA polymerase. We also call it as DNA dependent. We also call it as DNA dependent RNA polymerase. So it in prokaryotes, it has only one enzyme, that is known as RNA polymerase. It has only one enzyme, that is the RNA polymerase, and this RNA polymerase is also termed as DNA dependent RNA polymerase. Now why it is being known as DNA dependent RNA polymerase? That means this RNA polymerase individually cannot synthesize the mRNA. It needs the DNA strand, that means it needs the template strand to read and add the ribonucleotide in the complementary fashion. Now let's talk about the eukaryotes. When we talk about eukaryotes, in eukaryotes we can find three types of RNA polymerases. In eukaryotes we have three types of RNA polymerases. We have three types of RNA polymerases that are involved in the process of transcription. So first type is the RNA polymerase 1. RNA polymerase 1. This RNA polymerase 1 is basically used in the synthesis of the ribosomal RNA. This is basically used in the synthesis of rRNA. And this is the first type of RNA, and the second type of RNA polymerase is RNA polymerase 2, and this RNA polymerase 2 basically synthesizes the mRNA. What is the function? Its function is that it synthesizes mRNA or HNRNA. And the last type of the polymerase that is utilized there in the process of transcription of the eukaryotes, that is termed as RNA polymerase 3. That is termed as RNA polymerase 3. Now this RNA polymerase 3 is basically utilized in the process of the tRNA synthesis. So this is utilized in the process of or the synthesis of tRNA. So the raw materials that are required for the process of transcription is basically the enzymes and the ribonucleotides. So firstly, we have studied the enzymes that are required for the transcription. Now if you see in books, there is quite mingled that they have given that eukaryotes and prokaryotes, they have given mixed there the enzymes. But here first what we have done, we have written the transcription and we have given the enzymes both of prokaryotes and the eukaryotes. If you see in prokaryotes here, RNA polymerase, it's only one RNA polymerase, and this is DNA dependent RNA polymerase. And here again three types of RNA polymerases are being used. So this type of RNA polymerase is in eukaryotes. This is also DNA dependent. This is not DNA independent. This is also DNA dependent. So when we talk about the eukaryotes, we say that eukaryotes have three types of RNA polymerases, and that It has RNA polymerase 1, but once again this is also DNA dependent. So we say it has DNA dependent RNA polymerase 1 that is capable of synthesizing the ribosomal RNA. Then it has RNA polymerase 2, but this is again DNA dependent. So it helps in the synthesis of the mRNA. And it has RNA polymerase 3, it helps in the synthesis of the tRNA. This is clear. Now, while talking about the structure of this RNA polymerase, the RNA polymerase, when we talk about the structure, of RNA polymerase of the prokaryotes, or let's say about the bacteria. Let's talk about the bacteria. In this, we can see the structure of the RNA polymerase has 5 subunits. So we say that here in prokaryotes this RNA polymerase is being composed of 5 subunits. So it has 5 subunits. It has 5 subunits, and each subunit has its individual functions. So let's name the subunits. So when we talk about the first subunit is known as the sigma factor. So the first subunit is known as the sigma factor, and this sigma factor, the function of the sigma factor is basically to recognize the promoter region, to recognize the promoter region, So this is the first subunit, and when we talk about the second subunit of the prokaryotes, the second subunit is termed as, it has 2 alpha subunits. It has 2 alpha subunits, and these 2 alpha subunits basically help in the initiation, or they bind to the regulatory proteins, or their function is they bind to the regulatory proteins. They bind to regulatory proteins. So we are talking about the structure of the RNA polymerase. So first structure, as I said, that RNA polymerase is composed of 5 subunits. So the first subunit was the sigma factor, that the function of the sigma factor is that it locates or it recognizes the promoter sequence. Now the second subunits, basically they are the 2 alpha subunits. What is their function? They bind to the regulatory proteins. And now third subunit is basically known as beta subunit, and this beta subunit basically binds to the DNA template. This beta subunit, it binds to DNA template. It binds to DNA template. It binds to DNA template. When? That means during the process of transcription, it binds to DNA template. When this DNA template is being synthesized, this beta subunit holds it. It holds it, and then fourth is the beta dash subunit. This beta dash subunit, the function of this beta dash subunit is that it helps in the polymerization, or it adds the nucleotides. It adds to nucleotides. It adds to nucleotides. It adds nucleotides. Now, when we talk about, so in general, if we see that the subunits, overall subunits that are involved or that make the RNA polymerase, it has a sigma factor, it has 2 subunits of alpha, it has 1 subunit of beta, and it has 1 subunit of beta dash. And if we see overall, they make 5 subunits. They make 5 subunits. Now, if we see these 3, when these 4, that means these 2 subunits and this 1 beta unit and this beta unit, they are together and they make the enzyme that is known as core enzyme. They make the core enzyme. But remember, this core enzyme has the property of it can add there in the polymerization, but it cannot locate, or it cannot, this core enzyme cannot locate this promoter region. So for this, there should be this sigma factor attached to this core enzyme, and with the help of this sigma factor attached to the core enzyme, this RNA polymerase can bind to the promoter region. So once again, if we say that this is the RNA polymerase, this is the RNA polymerase, this is the RNA polymerase core enzyme. But remember, this RNA core enzyme or this RNA polymerase core enzyme cannot or is not able to recognize the promoter region for the efficient transcription. So what happens? It becomes active and the sigma factor becomes attached to it. And when sigma factor gets attached to the core enzyme, when this sigma factor gets attached to the core enzyme, what is the result? The result is that, see, I will write it, I will simplify it. We say that the core enzyme, we will write the core enzyme plus the sigma factor, plus the sigma factor. It makes the functional RNA polymerase. It makes functional RNA polymerase. It makes functional RNA polymerase. Now this functional RNA polymerase has a capability, it can join to the promoter sequence. So this was about the RNA polymerase of the prokaryotes. Now talking about the eukaryotes. In eukaryotes, as I see, or when I talk about eukaryotes, in eukaryotes, RNA polymerase does not have sigma factor. It does not have sigma factor. It does not have sigma factor. So, as you know that core enzyme without sigma factor cannot locate the promoter sequence. So, how does this RNA polymerase here in eukaryotes bind to the promoter region? So, here in eukaryotes you have the transcription factor second and transcriptional factor third. So, in place of sigma factor, here in the eukaryotes it have transcription factor second and transcriptional factor third. So, this basically TF stands for transcriptional, this is transcriptional factor. So, here in eukaryotes this RNA binds with the transcriptional factor and the RNA polymerase gets activated and it just binds to the promoter sequences. So this was the difference there in the RNA polymerase of the prokaryotes and RNA polymerase of the eukaryotes. Now let's talk about this transcriptional unit. Now as I told you about this, the first unit, this region, or this basically is whole the transcriptional unit. This is the transcriptional unit. This is the transcriptional unit, and this transcriptional unit, basically first region is the promoter. Now, what is basically the promoter? Promoter is basically the binding site of RNA polymerase. This is basically the binding site of the RNA polymerase. And this promoter region has certain sequences, so it has certain sequences like TA, TAA, TA. So it has certain nitrogen sequences or nitrogen bases sequences. So these are this is rich in these type of sequences. So this region is also known as Tata box. This is also known as Tata box. Now, why this is known as Tata box? Because it has certain sequences that are repeated continuously, that is TA, TAAA. So that is why this promoter region is also termed as Tata box, and this Tata box or these sequences were basically discovered by a scientist, and his name was Pribnow. And this Tata box is sometimes also referred to as Pribnow box. This is also referred to as Pribnow box. This is also referred to as Pribnow box. So, promoter, what is the function of the promoter? Promoter is the binding site of the RNA polymerase. So, if you see this promoter has certain sequences. It is rich in certain sequences. That means these sequences are TA, TA, TA. So, that is why this is known as Tata box, or this is also known as Pribnow box. Now, why this is known as Pribnow box? Because this was discovered by a scientist and his name was Pribnow. And now after that, we can see that this is the promoter region, and if we try to give the location, or when we write the location of this promoter region, we say that this promoter region is on the upstream, or we say this is, because when we write the sequences, we say that it is upstream, upstream 5 dash. So when we write the polarity, so we give it with reference to the non-coding strand. So this is at the 5 dash upstream of the non-coding strand, and after that you have this structural gene. This is the structural gene. This is the structural gene, and this is the terminator. So, when we talk about the structural gene, again, now this was the promoter, and now we talk about the structural gene. As I told you, that this structural gene basically gets transcribed, and this structural gene has the information for the polypeptide synthesis, or for the mRNA synthesis, or for the tRNA synthesis. So when we talk about structural gene, in case of prokaryotes, this is the structural gene is polycistronic. The structural gene is polycistronic, and in case of eukaryotes, this structural gene is monocistronic. This is monocistronic. So polycistronic means, in case of eukaryotes, it has lot of cistrons. That means 1, 2 and 3. That means simultaneously when here mRNA will be transcribed, and mRNA, after the process of translation, it will synthesize 3 or 4 enzymes from a single mRNA. But here in case of eukaryotes, we don't have these many cistrons. It has only one cistron. So that means when the mRNA will be transcribed, at that time what will happen? During the process of translation, only one polypeptide will be synthesis. So this was about the structural gene. The information present in the structural gene. Now the information present in the structural gene is on the non-coding strand. So this non-coding strand is basically read by the RNA polymerase, and when this RNA polymerase reads this strand or reads this strand, it adds the nucleotides. Now this is the terminator region. So when we talk about the terminator region, this terminator region basically contains stop codons. This terminator region basically contains stop codons. So that means these stop codons help in the removal of the mRNA transcript from this terminator sequence. So now let's talk about the transcription there in the prokaryotes first. So let's talk about the transcription there in the prokaryotes. As I told you that this promoter region is upstream at the 5 prime, because whenever we give the location, we give the location in contrast with the, or we give it with reference to the coding strand, and the terminator is basically downstream. It is at the coding strand, 3 prime downstream of the coding strand. So let's talk about the process of transcription there in the prokaryotes. Now initially when the prokaryotic transcription begins, what happens here? The RNA polymerase along with this RNA polymerase, the first step is the RNA polymerase along with sigma factor, binds to the promoter. So in case of prokaryotic transcription, when the transcription begins, what happens? This RNA polymerase, that means the core enzyme plus the sigma factor, they just bind to the promoter region. Now let's draw this promoter region. Let me draw it. Now suppose this is the DNA. This is the prokaryotic DNA. This is the prokaryotic DNA, and what happens during the first process? The first process is that RNA polymerase will come and it will bind. Now this is the RNA polymerase along the sigma factor. So in first process what will happen? RNA polymerase will bind. RNA polymerase will bind. RNA polymerase binds to DNA. RNA polymerase binds to DNA. Now after this RNA polymerase will bind to the DNA, what will happen here? With the help of the helicase, these hydrogen bonds in between the 2 strands of the DNA will be broken. Now see, as I told you that this is the 3 strand because this is the 3 strand because this acts as an template. This is the, and this is the 5 prime 3. So this binds to the strand which has the polarity 3-5 5 prime. So now the RNA polymerase binds to it, and second step will be that helicase will unwind or break the hydrogen bonds, will break the hydrogen bonds, will break the hydrogen bonds. Once the hydrogen bonds are being broken, the 2 DNA strands will be exposed. Once these hydrogen bonds are broken, the DNA strand will be exposed. Now, as we see that RNA polymerase binds to this strand, and here you can see now the DNA or in DNA molecule, these hydrogen bonds are being broken. These are the hydrogen bonds. Now what happens here? The DNA will unwind or break, or it will unzip. The helicase will unzip the DNA strands and it will expose the 2 DNA strands. Now suppose this strand, it has the polarity 3-5 prime. What will happen here? The enzyme RNA polymerase, what happens in the second step? In second step, RNA polymerase along with sigma factor initiates transcription. Once the transcription is being initiated, what happens? Once transcription is being initiated, now suppose now transcription is initiated, now in third step what will happen? This sigma factor will immediately get detached from the RNA polymerase. Now in third step what will happen? Sigma factor will get detached. Sigma factor will get detached from RNA polymerase. Now, after the attachment of the RNA polymerase along with the sigma factor to the strand, once the initiation has started, then this sigma factor gets detached from the RNA polymerase, and this RNA polymerase starts transcribing this structural gene. Now this is the RNA polymerase, this is without sigma factor. So now this RNA polymerase has the polymerization activity. Now as you know, this whole process of transcription in prokaryotes is taking place in cytoplasm. And in cytoplasm, when we say about the ribonucleotides that are being utilized for the process of polymerization, they are present there in the form of monophosphate. Now, like I'll be telling you, this whole process is taking place in cytoplasm, and there the ribonucleotides, when we talk about the ribonucleotide, like it has adenine monophosphate, and in place of thymine it has uracil. It has uracil, uridine monophosphate, and it has cytosine monophosphate, and it has guanosine monophosphate. So firstly, these need to get activated and being converted into triphosphates. So firstly, we will activate them. So this activation is being done with the process or by the enzymes phosphorylase in presence of energy. So, these get converted into triphosphates. Up till now, they were just in the form of monophosphate. Now, they will get converted into GTP, UTP. It will be AT, it will be adenosine triphosphate, and it will be CTP. So, now these are the bases that are in the triphosphate form. Now, as I told you in my previous lecture that these bases which are in the form of the triphosphate, they serve dual functions. Now, what are the dual functions? The first function is that firstly they will help there in the process of polymerization. They will act as a substrate there, and simultaneously the first function is they act as a substrate, and simultaneously the second function will be they will be providing energy for the process of polymerization. That means when the ribonucleotides will be adding here, the energy provided for the addition and for the establishment of the bonds, these will be provided by the triphosphate forms. So, now coming back to the original structure, when we talk about talking about the transcription, what happens once the sigma factor gets detached? Now, the third step will be that RNA polymerase will transcribe the template strand of DNA, will transcribe the template strand, that is the non-coding strand, using ribonucleotides. Using ribonucleotides as raw material, Now, once this RNA polymerase reads the whole structural gene, it will make the mRNA transcript. So, once this RNA polymerase moves, now this is the RNA polymerase, this is reading this template strand. Once it reaches to this region, now this RNA polymerase has reached to this region, and we say now this region is basically the terminator region. This is basically the terminator region. Now, as you know, this terminator region basically contains the stop codons. That means it gives RNA a signal that it should stop transcribing this strand. So, when this RNA polymerase reaches to the termination, so the next step will be, the fourth step is after transcribing the template strand, RNA polymerase reaches to terminator region. Once it reaches the terminator region, as I told you that this terminator region contains stop codons. This contains stop codons. Now, once this RNA polymerase reaches to the terminator region, here another factor that is known as rho factor. we write this rho factor like this. So this rho factor gets attached to this core enzyme of RNA polymerase. So now this rho factor gets attached to the RNA polymerase. So after transcribing the template strand, RNA polymerase reaches to the terminated region, and what happens there? Rho factor gets attached to the core enzyme of RNA polymerase, to the core enzyme of RNA polymerase. Now once this rho factor gets attached to the RNA polymerase, the transcribing activity of the RNA polymerase is being ceased. That means that RNA polymerase is now not able to transcribe, it stops. It stops here. Once now rho factor gets attached to the RNA polymerase, what happens? The transcribing activity of RNA polymerase stops. What happens? With this, the mRNA that has been synthesized gets detached. As a result, what happens? mRNA gets detached. mRNA gets detached. mRNA gets detached. The mRNA transcript gets detached. So that means now once this rho factor gets attached to it, what happens? The mRNA transcript that has been produced, this mRNA transcript, it gets detached, and RNA polymerase also gets detached from the template strand. And RNA polymerase also gets detached. So once this RNA polymerase is being detached, what we get is the, see, as I told you, now rho factor gets attached, and once the rho factor gets attached, this RNA transcript, this is after the attachment of the rho factor, this mRNA transcript gets detached. This is mRNA, and once this mRNA gets detached, then automatically what happens? This rho factor also gets detached. This rho factor also gets detached. This rho factor gets detached, and RNA polymerase gets detached, and RNA polymerase gets detached. Now once this RNA polymerase gets detached, and what happens here? Then the another enzyme that is known as the RNA ligases come, and DNA ligases here, in case of this double-stranded DNA ligase will come and it will join the template strand back into the position, into the original position. That means the hydrogen bonds will be established with the function of the ligases. It will join the two strands, the complementary base pairing will be established, and the hydrogen bonds will get developed. And this is the strand. What happens to this mRNA after the Now once transcription is over, that means the process of the transcription is over, at the end we got this mRNA. Now in case of prokaryotes, you should remember, in case of prokaryotes, this mRNA is being synthesized in cytoplasm only, in cytoplasm only. So immediately once this mRNA is synthesized, no modification takes place in this mRNA. So we say that no post-transcriptional modifications takes place. So in case of prokaryotes, no post-transcriptional modifications take place, takes place. It directly starts synthesizing the proteins or polypeptide. It directly synthesizes the proteins. It directly synthesizes the proteins. Now in prokaryotes, after the process of transcription, the mRNA transcript, it never needs any post-transcriptional modification, because this is polycistronic and this directly is a functional mRNA. So no changes in this mRNA, it directly goes there to the ribosomes and it synthesizes the proteins. So you have seen transcription in case of the prokaryotes. Now when we talk about the eukaryotes, the process is same, but somewhere there are some minor differences which takes place there in the process of transcription. And let's see those changes. Now in case of eukaryotes, now when we talk about the eukaryotes, now talking about the eukaryotes, now when we talk about eukaryotes, as I said that sigma factor is not there, so RNA polymerase, it just binds with the transcription factor 2 and 3. Now in case of eukaryotes, RNA polymerase 2 binds with transcriptional factor 2 and transcriptional factor 3. So here once again, this is the RNA polymerase in case of eukaryotes, and here sigma factor is not required, sigma factor is absent there in eukaryotes. So this RNA polymerase along with transcription factor. So here you have RNA polymerase, this is RNA polymerase 2 plus transcription factors. So once these RNA polymerase and transcriptional factors, they just bind to the template strand. They bind to template strand. They bind to template strand. They start the process of the polymerization. Now once again, after the binding, what happens there? The hydrogen bonds between the two complementary strands of the DNA, they get broken with the help of the helicase. So what will be the first step will be that RNA polymerase along with the transcriptional factors, they just bind to the template strand. Once they bind to the template strand, second will be that helicase will unzip. Helicase will unzip the two strands. Once helicase will unwind or unzip these two strands, now this strand that needs to get transcribed will get exposed. will get exposed. Now simultaneously, as you know, here once again the topoisomerases and the single stranded binding proteins, they will bind to this strand that needs to get transcribed and they will stabilize it. Once this is stabilized, what will happen now? This is the promoter. Once this RNA polymerase and transcription factor, they just bind to the promoter. Now they will move to the structural gene. Now this is the structural gene. Now once they move to the structural gene, they will read the sequences on this structural gene and they will polymerize. So they will add the RNA polymerase will go on adding the ribonucleotides. So once it gets on adding the ribonucleotide, it will form a mRNA. So once this reads, RNA polymerase will read the structural gene, it will transcribe it and it will form a mRNA. Now this is the mRNA. Once this has been read and it reaches to the terminator. Now same in case of that. See, first we will write here. Helicase will unzip the two strands. Then what will happen? RNA polymerase 2 will transcribe the template strand, Now here again, as you know, in case of the polymerizations, again ribonucleotides are being utilized. There in prokaryotes you have seen the activated ribonucleotides are used. Now what is the difference here in the polymerization of the eukaryotes and prokaryotes? If you see there in prokaryotes, this whole transcription took place in the cytoplasm, but here in eukaryotes this transcription is taking place in the nucleus. When we talk about the eukaryotes, we say the process of transcription there in the eukaryotes, it takes place in the nucleus as compared to the prokaryotes. If we see there in prokaryotes, the transcription took place in the cytoplasm. Now here is the difference there in the transcription of the eukaryotes, this whole process is taking place in the nucleus. So that means now the ribonucleotides that are used for the polymerization of for the polymerization of the ribonucleotides, these are present in the nucleus. So firstly, as in the previous step, you have seen that in nucleus they are present in the monophosphate form. Firstly, they need to get activated and they are being converted into triphosphates. So same process here. RNA polymerase second will transcribe the template strand using the ribonucleotides. Once again, RNA polymerase will come along with the transcriptional factors. It will join to the promoter sequence, and there you can see a small space is formed, that is known as transcription bubble is formed. When the transcription bubble is formed, what will happen? Then this RNA polymerase will move to the structural gene, and in case of eukaryotes, you can see the structural gene is monocistronic. That means only one cistron is present here. So this RNA polymerase has the polymerization activity. It will move towards the structural gene and it will synthesize the mRNA transcript. Now this mRNA transcript is being synthesized. Once this mRNA transcript is being synthesized from the information that is present in the template strand, and it finally reaches the terminator signal. Now when it reaches to the termination, there you know that it has this sequence that is known as the terminator. Now here again we don't have this rho factor. Here we have certain proteins also. Now these proteins, they will bind to the RNA polymerase. Once they bind to the RNA polymerase, what will happen? The DNA transcript will get detached from this DNA template. Now let's read it. RNA polymerase second will transcribe the template strand using the nucleotide. Now, once the structural gene is transcribed by RNA polymerase, it reaches to the terminated region. As you know, in terminated region, there are the stop codons. Now, this stop codons will stop the process of transcribing. Now, these stop codons, they will stop the process of transcription. Now, immediately what will happen? Once it reaches to the process, once it reaches to the terminated region, the process of transcription will start. The process of transcription, the process of transcription will stop. As a result, what we will get? We will get a mRNA, we will get a mRNA primary transcript, mRNA primary transcript, mRNA primary transcript, or this is also known as nascent RNA. This is also known as hnRNA. This hnRNA means heteronuclear RNA. Heteronuclear RNA. So, process of transcription will stop. We will get mRNA primary transcript. This mRNA primary transcript is also known as nascent RNA. This is also known as heteronuclear RNA, and this mRNA will be synthesized there in the nucleus only. So, along with this mRNA, we will get RNA polymerase. So, at the end of the transcription, once the transcription is over, what we get? We get this mRNA primary transcript and we get RNA polymerase. Now, let me show you diagrammatically. It is once again now suppose this was the area that was to be transcribed, and this was the terminator region and this was the promoter region. Now, once the RNA polymerase reaches here, that means at the end of the transcription, what happens? The mRNA that was being synthesized, it gets detached. So, you get this primary mRNA transcript, and along the primary RNA transcript, here was RNA polymerase, and you will get RNA polymerase. So, this is RNA polymerase at the end of the transcription, and this is the mRNA. Now, when we talk about this nascent RNA is not functional. This nascent RNA is non-functional in case of eukaryotes. Now, there in case of prokaryotes, you have seen that this mRNA that is being synthesized after the process of the transcription, it was there in the cytoplasm only, and simultaneously the process of translation started without any post-transcriptional modifications. Now, here in case of eukaryotes, this mRNA primary transcript is non-functional. This is non-functional. And why this is non-functional? And this is being synthesized in the nucleus only. So, after that, once we get the mRNA transcript, this mRNA transcript or the nascent RNA undergoes post-transcriptional modifications in the eukaryotes. So, once this nascent mRNA is being synthesized there in the nucleus of the eukaryotes, so this is the nascent mRNA that has been synthesized in the nucleus. that has been synthesized in the nucleus of the eukaryotes. But as soon as this nascent mRNA is being synthesized, this is non-functional. Now this is non-functional because of certain points. So firstly, in order to make it suitable for protein synthesis, this nascent mRNA needs to undergo certain post-transcriptional modifications in case of eukaryotes. As I have already said to you that in case of prokaryotes, it is functional as soon as it is formed. But in case of eukaryotes, there is something different. The difference is that once this nascent mRNA has been transcribed, it is not functional. It needs to undergo certain modifications to become functional. So these modifications are termed as post-transcriptional modifications. So in post-transcriptional modifications, what happens? The first procedure or the first step is that this nascent mRNA undergoes capping. Now what do you mean by capping? As you know that this nascent mRNA has two ends. One is the 5' and one is the 3' end. Now towards the 5' end an methylated guanosine cap is being added, or we say methylated guanosine triphosphate is added at the 5' end. So this is the methylated guanosine triphosphate cap is being added towards the 5' end, so that this transcript, once it travels from the nucleus to cytoplasm for protein synthesis, it will be protected. Now this 5' end will be protected from degradation of various various biomolecules there in the cell. Now after this methylated guanosine is added to mRNA there in the process of capping. This is the first step. Then after capping, second step is the tailing. In tailing, what happens? This towards the 3' end. Now in methylated guanosine cap, this was added at the 5' end. Now in the second process or in the second post-transcriptional modification, there and towards the 3rd end, adenylate residues, nearly 200 to 300 adenylate residues, they are added towards the 3' end. So we say that there is the addition of adenylate residues, that is, what is the number of the adenylate residues? Usually say that there are 200 to 300 are added towards the 3' end, or we simply say in tailing a poly A tail is added towards the 3' end. Now after that tailing, the next process is, the third process is splicing. Now once we talk about splicing, as I told you, you have this nascent mRNA transcript. Now this nascent mRNA transcript as a whole, I said it is non-functional. It is not because of the ends need to be protected. If we see this whole mRNA transcript, this is flanked by certain sequences that do not code for anything. They are known as non-coding sequences. So the coding sequences are in the form of exons. So when we talk about the coding sequences are in the form of exons. The coding sequences which code for a particular amino acid, they are in the form of exons, and certain sequences which do not code for anything, we call them as non-coding sequences. We call them as non-coding sequences. So these non-coding sequences are in the form of introns. So you should remember this nascent mRNA contains two type of coding sequences. One is the two types of sequences. One is the coding sequence and one is the non-coding sequences. So these non-coding sequences, as I said, in the form of introns, they do not code for anything. So before going to the cytoplasm, these non-coding sequences need to be excised from this nascent mRNA transcript. So this process or these introns are removed or excised by a process that is known as splicing. So what happens in splicing, these introns are basically at particular sequences, they are acted upon by an enzyme that is known as ribonuclease P. That is known as ribonuclease P. Now this ribonuclease P, it excises these two ends. That means from the beginning of the intron to the ending of the intron, it cleaves these two sides, and the structure of the intron which it forms after cleaving is termed as lariat structure. Is termed as lariat structure, or we also saw a spliceosome is formed. You can see these are basically the spliceosomes, and these spliceosomes are basically the excised non-coding sequences known as introns. So in the process of the splicing, we have seen that the non-coding sequences are being removed from this nascent mRNA transcript. So this was the third process, and fourth process is known as ligation. Now what after once these spliceosomes are removed by ribonuclease P, then you can find certain regions are there where these regions where from the spliceosomes have been removed. Now these regions need to be joined again. So this is an intron, this is an intron, this is an intron. Now in between two introns there is a gap. There is a gap. Now this gap needs to be filled, or this gap, the two exons need to be joined. So these two exons or these two gaps, they are joined by a process that is known as ligation. So this ligation is basically the joining of the two exons. When whole of these exons in an nascent mRNA transcript are joined, it forms now at the end of the ligation a functional mRNA is being produced. So this process of ligation is by a process that is known, now this process of ligation is done by an enzyme, or this filling of the gaps. Here an enzyme is used that fills the gaps or that joins the two axons. That enzyme is known as ligase. Now ligase helps in joining of the two exons. Now once these exons are joined, a functional mRNA transcript is formed. Now after this, now the functional mRNA transcript has been formed. Once this functional mRNA transcript is being formed, it is transported from the nucleus to the cytoplasm. So in eukaryotes, once again I will tell you, in eukaryotes what happens, that once the mRNA is transcribed, now this mRNA is not functional. This mRNA needs to go under certain post-transcriptional modifications. So this nascent mRNA, or we also call it as heteronuclear mRNA. Just remember, these are certain terms which you should remember, that this nascent mRNA or this heteronuclear mRNA, this needs to be modified first, because it is non-functional and it is susceptible when it will move from the nucleus to the cytoplasm, it is susceptible to various biomolecules. Maybe they might attack it and they might degrade it. For that it need to undergo certain modifications in order to become completely functional. So first modification that this nascent mRNA goes is capping. That means towards the 5' end of the nascent RNA, a methylated triphosphate guanosine cap is added, and after that what happens at second type is, or the second step is the tailing. In tailing what we do, that a poly A tail is added, and after once at 5' end methyl guanosine cap is added, and at the 3' end the polyadenylate residues are added, then it undergoes another process. That process is known as splicing. Now, what is basically splicing? Splicing is the removal of the non-coding sequences that are in the form of introns that need to be removed from the nascent mRNA transcript. So, these nascent mRNA transcript, once these introns are being removed, these introns are removed with the help of an enzyme that is known as ribonuclease P. It excises the introns, and in the form of the lariat structure, these introns move out. Now, the next step after the splicing is the ligation. Once the introns are being removed, in between the two consecutive exons, there is a gap, and this gap needs to be joined, or these two exons need to be joined. So, these two exons are joined with an enzyme that's known as ligase. So, ligase facilitates the the joining of the two exons. Now, once this ligation takes place, this mRNA transcript, this nascent mRNA transcript now becomes functional mRNA transcript. And now this functional mRNA transcript, it moves from the cytoplasm to the, it moves from the nucleus to the cytoplasm. Now, once this functional mRNA transcript has been produced there in the nucleus, so this is the functional mRNA, this is the functional mRNA. Now, in case of eukaryotes, this functional mRNA has been produced there in the nucleus. Thereafter, the after post-transcriptional modifications, now after that it is transported. Now, this functional mRNA is transported to cytoplasm. Now, it is transported to cytoplasm, and there inside the cytoplasm, it goes towards the ribosomes. It goes towards the ribosomes. Now, when this functional mRNA will reach the ribosomes, and then at ribosomes it will start synthesizing proteins. But once again, the information regarding the protein synthesis is there in the mRNA. There should be some molecule that will read the code present on the mRNA and will translate it into a polypeptide. So, that means when this will be transported to cytoplasm, and in cytoplasm it will go to ribosomes. Now, on the ribosomes, there you need a helper molecule, or you need an adapter molecule that will read the code on this mRNA transcript, and it will synthesize the proteins. So, this process is termed as translation. Translation is basically a process where a message is being read from the mRNA transcript and simultaneously protein is being synthesized. But once again I am telling you, you need a helper molecule that will read the message there in the mRNA and will translate it into proteins. So, we will be doing this in next lecture. Thank you.